Hello and welcome back still here in the Bella Center at the hallway studio of Transplant Live. If you're now watching this on Facebook and YouTube, it's glad, we are happy to host you here, but you should know that you're missing out on some of the best things because every session in the big rooms is being recorded and live streamed to ESOT Transplant Live. So if you haven't done so, hop over. Those who were there have just seen Plenary 2. If you're not, let's summarize. Because I'm now joined by Menna Clathworthy from Cambridge in the United States. You were chairing the Plenary 2 session and also one of the first speakers, which was titled Single Cells Omics, Generating an Atlas to Chart Transplant Organs.
Yeah.
In your first talk, you introduced the Human Cell Atlas. For our viewers who've missed it, what is that?
Yeah, so the whole session was how we might use omics technologies to better understand transplant organs and maybe even in the future to build transplant organs. So at the heart of that is the Human Cell Atlas project. So this is an international consortium, so a group of clinicians and scientists with various expertise that have come together with the aim of trying to generate a comprehensive atlas of every single cell, in every single tissue, in every single organ in the human body.
Wow.
And we kind of feel that by characterizing cells in normal tissues, we can then move on to understand how they change in disease, and that will give us insights into what we can do to prevent that. And so what I went on to do from there was just to describe a little bit about how much the Human Cell Atlas project has developed.
Yeah, because where are we? Is it, first thing I'm already blown, like, is this possible? Well, apparently, that's why you set out on the journey.
Yeah.
Right?
So we've been doing this, so the consortium came together in 2016, so we've been doing this for 3 years. But already it's expanded massively, so we have more than 1,500 members of the HCA consortium, and from more than 60 countries. And
who are they? These are researchers throughout the world sharing data?
Exactly, and that's really an ethos of the HCA project, is that we want to share data as quickly as possible. So generally we try and make all of our data open access so that other researchers out there can use the data to take the whole field forward. And so what's allowed this is developments in technology. So now, whereas previously we would generally, and in transplantation in particular, take whole tissue biopsies and have a look at the mRNA transcripts within the biopsy.
Yeah.
Now we can actually take individual cells, not just a whole biopsy, individual cells, and look at the messenger RNA transcripts. And that allows us to define sometimes new cell types that we didn't know about. So there was an example of that last year where in the lung they found a brand new cell type, the ionocyte, and that cell type actually, it turns out, expresses the cystic fibrosis protein.
Okay, so a very important marker.
Yeah, a very important marker. And then in terms of the kidney cell atlas, which I've been involved in building, we've actually been looking at kidney in human embryo, so to look at kidney development. And that gives us information that will be useful for building organs. So if we're trying to generate organs from stem cells from scratch, knowing the signals that allow cells to become differentiated cells within an organ is helpful.
So just to really understand, so you're literally researching how in an embryonic state organs develop from single cell multiply, and then which elements are built on top of which?
Yeah. So what we can do is take kidneys at different post-conception weeks, so from very early on, look at the cells and their transcriptome at that stage, and then compare that to a few weeks later. And then we can make a trajectory of cells and ask, how does their transcriptional state change as they become more mature?
Wow, very exciting work.
Yeah.
Now, obviously, people can go into the Transplant Live library and see the entire session.
Yeah.
Last question, if people are inspired by this cumulative effort, how can they step in and join the movement?
Yeah, so you can go to the website, so humancellatlas.org, and you can just sign up there
and start sharing your data.
Yeah, and then we're also trying to make our data available so that researchers out there can interact with it. So we've made a simple website, so Kidney Cell Atlas, that you can again log into, and maybe if you're interested in a particular gene and where it's expressed, you can look exactly where it is in the kidney.
Wow.
So yeah,
very
pretty
exciting work, and thanks for sharing that here in the Congress.